Afternoon, everyone. Very, very happy that you've joined us either in person here in the auditorium or else online. We've got a bunch, we're good to go online, Rebecca, right? Okay, awesome. We've got a bunch of people who have joined online to hear our amazing guest speaker today, Dr. Mike Murphy. Mike Murphy, who will tell us, I mean, you'll see his title in a moment, but you'll really understand the arc of the type of work that he does. But he's a current, I'll talk about where he's been, and then I'll talk a little bit about what he's done. He's originally trained at Trinity College in Dublin, and then he did his PhD in Cambridge. No big deal, Trinity College in Cambridge, not, haven't heard of them. He's trained in the United States, in Zimbabwe, Ireland. He eventually took up a faculty position at the University of Otago in New Zealand. And in 2001, he moved to the MRC unit, the mitochondrial biology unit at Cambridge. And that's where he currently is a program lead. He's a professor in the mitochondrial redox biology, and a Wellcome Trust investigator, an MRC investigator, an honorary research professor at the University of Otago in New Zealand, a recipient of the Kylan Medal from the Biochemical Society, an honorary fellow of the Royal Society of New Zealand, and a fellow of the Academy of Medical Sciences. He's published over 400 peer-reviewed papers, more than 74,000 citations, and he has an age index of 139. Most importantly, beyond all of those accolades, which are very well deserved, Dr. Murphy is really a perfect example of a translational biologist. His research focuses on the roles of reactive oxygen species in mitochondrial function and pathology. So as we know, reactive oxygen species, I was just telling Dr. Murphy that I teach my students, they're not just the bad guys. We hear about them a lot as bad guys, but in fact, they do a lot in the cell, both good and bad. His research has pioneered the targeting of these bioactive probe molecule to probe mitochondria in vivo. We now use many of these reagents, such as things like MitoQ. Many of you have probably heard of it. We now, thanks to Dr. Murphy's work, such as MitoQ, there's lots of other work in the pipeline that can protect against oxidative damage in things like what we're going to hear about today, ischemia-reperfusion injury. Dr. Murphy and his colleagues actually developed this into an oral drug that went to phase 2 trials, and actually used this as a sort of proof of concept that this type of biology, this type of signaling can be used for therapeutics, which has really opened up the field and allowed us to understand that these signaling pathways in mitochondria can be relevant therapeutic targets. The Murphy group has now created I don't know if you pronounce it MitoSNO, but MitoSNO, mitochondrial targeted nitric oxide donor, and MitoG, and these can be used to treat diabetes. Now we know from this work is extended to the fundamentals of how mitochondria produce these free radicals during ischemia-reperfusion injury. And this is really allowing us to be able to translate some of these findings as he's done with other diseases. So Dr. Murphy's work has really, again, it's the arc of discovery and understanding not just the fundamental science, but with the lens of how can we target it? What are the principles of targeting it? And what do we need to actually move this into the clinic? So with that, I welcome Dr. Murphy to the podium. Thank you.
Well, that works. Oh, there we go. Oh, right. Okay, so what, in my view, the heart is just a great big bag full of mitochondria. So I'm only thinking about it in terms of mitochondria. But we're trying to understand what goes on. And what we discovered during ischemia is that the mitochondrial metabolite succinate accumulates. What happens during ischemia, when it's anoxic, so the electrons have nowhere else to go. They pile up on a few substrates, in particular, they pile up on malate in the cytosol, comes into the mitochondria, goes to fumarate, and it dumps the electrons on the cube from the cupule, come along, reduce fumarate to succinate. But succinate has nowhere else to go, so it's a dead end, like lactate would be in an overworked muscle. So what happens is the succinate builds up dramatically during ischemia. And we can show this here in a model that we use quite a bit, LAD ligation model of a mouse heart attack. So what happens here is this mimics a situation of having a heart attack. So what happens is you tie off a LAD here, you tie this off, the area downstream of that becomes ischemic. And then you can untie this and reperfuse that area of the heart. And this mimics what would happen if you had a heart attack. You went into the cath lab and they removed the clot and reperfused the heart. So those situations would be what would be happening here. And you can sew the mouse back up again and you can measure the heart function weeks or months later. So when we do this, what we can do is that we can actually look at the succinate distribution inside the heart. We use a technique called imaging mass spectrometry here. So you freeze the heart and take a slice through the heart. You raster across it with a beam and you puff up the metabolites and you can measure the amount of succinate that's distributed through the heart. And here you can see with the false color here, you can see we're getting this dramatic accumulation of succinate in the infarct zone here. So you get this local area that's infarcted, you get this dramatic accumulation of succinate. The important thing, though, is that when you reperfuse the heart, the succinate goes away. Here you can see the infarcted region. So different heart we've measured after reperfusion, you can see that we've now lost the succinate. So the idea was that the succinate is somehow storing the electrons. We can confirm this a bit later here. If we look here, we smash up the heart and measure the succinate by mass spectrometry, LCMS-MS, and you can see the succinate accumulates dramatically and then goes back down upon reperfusion, goes up, back down upon reperfusion. So the idea was that the succinate is storing the electrons, and those electrons are then driving the ROS production upon reperfusion. So how could succinate be doing that? Well, it was known a long time ago in the mitochondrial field. There's a process called reverse electron transport. And what happens there is that the electrons that are in succinate can get pumped back into the respiratory chain here, oxidized by succinate dehydrogenase, and highly reduce the cupule. This pushes electrons down through complexes 3 and 4 to oxygen when the oxygen comes back in at reperfusion. And you pump out protons building up a very high protomotor force. The reduced cupule and the high protomotor force cause complex 1 to go backwards. So that's reverse electron transport through the iron sulfurs onto the flavin mononucleotide here on the surface of the complex one. And from there, the electrons can react with oxygen from superoxide. So this, we propose then, is this using up the succinate and driving this burst of ROS production at reperfusion. So how can we explore that a little bit in the model we looked at there in the mouse? So if we look at the LAD model, go back to that, and we want to see, could we actually measure ROS production in the mouse in situ? There are lots of methods to measure ROS using fluorescent probes and things, but you can't use in vivo. Doing these ex vivo is not ideal because you need to actually have the conditions inside the heart, the calcium, the ATP, the oxygen tension, the substrates, etc., have to be the same as they are in there. And it's very difficult to mimic those precisely in a cell system or something like that. So we want a method to measure ROS inside the heart here. So when we're measuring ROS, it's an interesting area. It's a bit like the ancient explorers when they were lost in the ocean. Instead of saying, we don't know what the hell's going on or where we are, they say, here be dragons, give the impression that they have some idea what they're doing. When in reality, in ROS field now, a lot of people will just say, here be ROS, when they're saying they don't understand it, or the reviewers want to confuse the reviewers who won't understand it either. And so they can bypass the criticism that way because nobody really understands ROS. We're trying to go beyond that now. What we're trying to do is see, can we actually set up probes to measure ROS production in vivo, in the living animal? This would use a system of phosphonium targeting molecules like this. What we know is that mitochondria have very high membrane potential at 180 millivolts. Doesn't sound like a lot, but you can't actually put a potential any bigger than that across a membrane without it undergoing dielectric breakdown. So what we can do is we can use this as a target group, and we can tag onto this various things, such as mitoQ or whatever. In this case, we call it mito B, because everything targeted to mitochondrial mito, and we call it B because we've got a boronic acid moiety attached here. The notion with this is you inject that into a mouse, it will go into cells very rapidly, into mitochondria, and accumulate essentially 1,000-fold into the mitochondria because of the membrane potential, which means as far as you're concerned, it's only in mitochondria. So if you take it out of the take out the heart of the mouse, you're reporting what's going on in the mitochondria. This is where the boronic acid comes in. The boronic acid will react selectively with hydrogen peroxide, also with peroxynitride, to form a phenol. And so the more hydrogen peroxide in the heart inside the mouse, the more of this goes to this. Because they both accumulate to the same extent, the ratio of P to B will be high if there's a lot of hydrogen peroxide and be low if there's not much hydrogen peroxide. So we can measure this ratio ex vivo by LC-MS, very sensitive down to picomole levels relative to deuterated internal standard. So we can now see, how much exposure to ROS there was in the heart under those conditions. So we move this on then, we can look in the heart here, and we can see the mito P to mito B ratio here is very low in the normal heart, but when we reperfuse, it goes shooting up. So now we've confirmed there's ROS production in the mitochondria upon reperfusion. That was sort of assumed, but we want to see, because that actually being driven by succinate oxidation. So how are we going to investigate that? What we can do is we can block succinate oxidation production by succinate dehydrogenase by using a very simple inhibitor, malonate. Obviously, malonate won't get into cells, it's got two negative charges here. So we use a dimethyl ester, which is a liquid, and it will go straight through membranes into the cytosol, and esterase will break it down, and then it will block succinate dehydrogenase. So if we pre-treat the mice with dimethyl malonate, and then let's see what happens. What you can see under those conditions, where we block the succinate accumulation, what you can see here is that in the control, it's low, reperfusion, it's high. If we pre-add dimethyl malonate, we block this oxidation event here. So that's consistent with the succinate driving the ROS production. But if the ROS is causing damage, then by blocking this succinate oxidation, ROS production, we should also protect the heart against the reperfusion injury. So we can see here, if we do histology on the heart, what you can see is you've got this area is the dead tissue. It's less than the dimethyl malonate treated heart. And here we quantify this over a range of different hearts. You can see we drop the dead tissue from 40% of the area at risk down to about 15% here. So we're protecting the heart from that ROS production there. The next thing we want to do then is to see, well, if that's consistent with succinate accumulating, being oxidized quickly and driving this production of ROS, then how can we be certain that it's RET? Because this is quite a convoluted little process here. We've shown succinate is driving it, but maybe it's from some other site. You can infer that it's complex 1 RET indirectly, because what you can do is you can add inhibitors of complex 1, such as rotenone, and you would block this ROS burst. Or you can uncouple, which prevents RET occurring, and also block the ROS burst there. So those things, when you do those things, then what will happen is you will prevent some of this ROS burst. So that's consistent with it being complex 1. But to do a more direct test, we're very lucky in the sense that there's a mutation in mitochondrial DNA. Remember, mitochondria have their own DNA. There's a mutation in mitochondrial DNA that introduced a single amino acid substitution, the ND6 subunit of complex 1. And what happens there is that you get a single amino acid substitution in the ND6 subunit in the core of complex 1. And Doug Wallace's lab, who discovered this mutation, when they make mice contain this, when they have mice that contain this mutation, they suggested that there was no reverse electron transport in the complex 1 due to this point mutation. So we could confirm that if we, and Doug very kindly gave us those mice. What we could see here was that you can see that in the isolated mitochondria, we're getting a lot of hydrogen peroxide here, production from when we feed them with succinate. But in the ND6 mice here, we're not getting this hydrogen peroxide production. So there's no RET occurring in these mice. Then we take those mice and we do the same as before. We use the mito-P ratio measurement. And what you can see here is in the wild type mice, you're getting this burst of ROS production upon reperfusion. But in the ND6 mice, which don't have RET, you're not getting this burst of ROS on reperfusion at this point here. So that's all consistent then. Oh, and also the damage to the heart. The large infarct in the wild type, a lower infarct in the ND6, as you can see here. So these are all consistent then with the idea that we have certain oxidation driving high protomotor force and RET at complex 1. But the other point is that what we're saying here is that this occurs very early in reperfusion. So as soon as you turn the oxygen back on, what happens is that the RET occurs within the first minute or so, when the succinate is oxidized. So what that implies then is if RET is the process, then it says the protomotor force, the membrane potential, has to switch back on very rapidly indeed, within the first minute or so. So in other words, the mitochondria inside the heart have to go from being ischemic for about 20-30 minutes to being fully active in a few seconds. It seems like a big ask. So we want to see, is that actually occurring? Now, how are we actually going to measure membrane potential? Well, that's very challenging to do. You can do it by the distribution of things, a bit like the mito-B, but the rate of accumulation and the quantification of that is quite challenging. And also, it's very hard to visualize those things inside a heart. You can use fluorescent probes as well. But again, visualization is challenging, and the timescale means you can't really do this. So very fortunately, I managed to collaborate with Bob Balaban of the NIH and Abigail Giles, a very good PhD. She can just finish her PhD. So she did, working with Bob, she came up with a method. Well, Bob's been working on this stuff for 30, 40 years. And from that, what we have is a way to actually try and measure the membrane potential inside a perfused heart. What we do is we use an endogenous chromophore that's membrane potential sensitive. And it turns out, now you remember at your mother's knee, you learned about the Q cycle and all those sort of things. Well, the Q cycle requires electrons to distribute on the BC1 complex or complex 3. The electrons move between two B heaves, cytochromes, a low potential and a high potential. That's the way the electrons moving down the spiritual chain, that's how it's converted into a membrane potential by moving this electron back and forth, or how it sets up the charge from chemical to electrical energy. What happens is the low potential electron, a single electron is shared between these two heaves, and it'll move back and forth depending on the membrane potential. So if there's a high membrane potential, the electron will tend to be moving here onto the low potential heme. It's confusing. The low potential just refers to the electrical potential because it's more reducing. And the high potential heme will tend to take the electron under low membrane potential as the electron gets sucked onto this one here. Okay, so what you can see then is the distribution of the electron is proportional in some way or relates in some way to the membrane potential. And we can measure this because if we take the spectra of these, then the spectra will change whether it's reduced or oxidized. So if we measure the spectra of these guys, then we can infer the membrane potential. Easy to say, quite difficult to do. So what happens is that Bob and Abigail set up this system where you take a mouse heart in a Langendorf type system, and there we put a light catheter through into the left ventricle, and the light comes shining through. And we're just doing a spectra of the heart here. Technically, it's quite demanding. So what you do here is you have the heart hanging inside this chamber, and then you're trapping all the light in this sphere, which focuses the light back onto a single light guide, and then you have a spectrometer here. So you can measure a spectrum of the light as it's passing through and scattered through the heart there. What we do then is we do a bit of very fancy stuff that Bob came up with. So this is the raw spectrum. Well, it's not quite the raw spectrum. This is the spectrum over this area of interest where the hemes absorb and all the other cytochromes and things are absorbing here. So you can see the spectrum here. What we do is we break that down into a series of reference spectra, and we just add these up till it generates something like that. And that tells us what the spectra of these guys were. And from that, we can infer the reduction state of heme B and heme L, as well as cytochrome C1 and C. And then we can just show how good it is by subtracting this from that, and it shows that the residuals are very small. So we've got an accurate image then of the spectra of those things inside the heart. Now, how are we going to go from that to try and measure the membrane potential? Well, we can generate a derived parameter here, which is just the absorbance of the low heme against the total here. So this absorbance here shows, this gives you the amount of reduced BC1 complex. So there's a single electron either on or off the complex. So here we're only looking at the B hemes that contain an electron, and the ratio of this to this tells you the proportion that is stuck on the low or not. There. So FBL, you can see in isolated mitochondria, with succinate it's high, nitroisomate it's high, with malonate it's low, with ADP it's lower, with non-coupler it's far lower. So you can see there's a relationship between a high potential and a low potential and this number here. But to calibrate that, what we do is, again, we measure the membrane potential by the distribution of a cation, TPMP, with an electrode, and in parallel, we also measure the FBL by the absorbance. We do the experiment inside the sphere, we have the electrode and the light capture at the same time. And here what we do is we titrate the membrane potential. What you can see is the membrane potential going from low to high. We get a sigmoidal thing, and we can fit this curve with this equation here. And so if we measure FBL in the heart, we can read off the membrane potential here, okay? So doing that, then we go back to our experiment. And look what happens here if we have the heart on its own inside here, and we can equilibrate it for 20 minutes. We can expose it to ischemia, and then we can reperfuse. And because we're taking a spectrum every second, and we can then do the ratio of the B and L hemes when we do all the correction, we can measure the membrane potential every second right through this process here. Remember, you may have forgotten by now what the original point of this was. The original point was what's happening on reperfusion. Is the membrane potential coming back quickly or slowly? It has to come back quickly if RET is the mechanism. So here's what it looks like when we perfuse the heart. Here's the membrane potential during ischemia, during normoxia. Here's ischemia, so it drops down. Now it drops down because there's no oxygen, so the proton pumping by the respiratory chain is not happening. But it's being maintained now, we think, by the ATP synthase going in reverse, pumping protons backwards to set up a membrane potential. And then what happens at this stage is this is hypercontraction. The ATP levels drop, they're being generated from glycogen and glucose at this stage, and then the ATP runs out, the potential collapses at this stage. This is where we're reperfusing, and you can see the potential goes shooting right back up to the maximum at this stage here, and then fiddles around a little bit. We won't go into this at this stage, but it could be the permanent transition point and things which we'll talk about later. But this is the bit we're interested in, so let's expand that. And what you can see is that as soon as we reperfuse, the potential goes shooting back up within a minute. So this is consistent with that potential being able to drive RET at complex 1. So that's addressed the main question. We could also measure the succinate levels in the heart as we're reperfusing it. And now you can see here that the succinate is very high at the start, and after a minute, the succinate has gone back down. So the succinate oxidation is driving the re-establishment of the membrane potential on reperfusion. So that's what we think is happening. And so what we have then is we think a complete description of ischemia-reperfusion injury, which applies to the heart, but all other organs as far as we know. So this was, a lot of people were speculating on what was the cause of ischemia-reperfusion injury. And at the time, it was thought to be a fairly random process where electrons would just jump off any reduced site, produce a ROS, and that would be damaging in kind of a vague, uncharacteristic way. But now we know a couple of things. We know exactly the process, and we also know the timing is occurring in the first few minutes of reperfusion, which also explains why so many drugs never work, because they're happening. If you're giving the drug 10 minutes later, it's all over. So now, having understood the process, we want to try and see, could we actually drug the process? Obviously, what we've said is that if succinate oxidation is driving this ROS production, then we should be able to block succinate dehydrogenase with malonate, as we showed before. And if we do that by giving a dimethylmalonate to get into the cell easily, then it'll produce malonate locally, as this will be the prodrug, this will be the active drug, and then when it inhibits succinate dehydrogenase there. And we showed before that this is protective. Okay. So why don't we see if we can use dimethylmalonate as a potential drug to stop this? So let's consider how we might do that in a patient. That's a patient. And what we're doing here is PPCI, or primary percutaneous coronary intervention. This is what they do in the cath lab. So if you get a heart attack, they'll wheel you in, they'll do a CT scan or whatever, and they'll try and place this through large blood vessels in your groin, go right up through here, in there, and pull out the clot, and then you'll be reperfused in that area inside the heart at that point there. Okay. So that's what happens upon reperfusion injury. So what we want to do is try and see, at that stage, could we actually use dimethylmalonate? And that, this is essentially the model of, the mouse version of this is what we're doing the modeling. So let's just see what happens here. This is the timing of what happens. So this is pneumoxia. This is you sitting at home or on a Peloton bike or whatever, and suddenly you get a heart attack. And this is the ischemia thing. You want to get that as minimized as possible. So you're going to be blue lit to the hospital, and then you're going to have the PPCI here. So unless you plan your heart attack really well, the only time you can invite the therapy is here. If you're planning a heart attack next week, you can start taking either Q or various drugs, and the outcome will be far better. But in most situations, you're only going to be able to treat the heart attack at this stage here. So this would be when you add the drug. So we can add the drug at the same time as we're reperfusing the heart. As soon as you're putting out the clot, you want the drug in at that stage. Remember, it has to be, it can't be given 10 minutes later. It has to be given at that time while the succinate is being oxidized, in our opinion. So let's see what happens. Can we use dimethylmalonate as a good drug for this? So what you can see here is dimethylmalonate with the two methyl groups here gets into the cell. If we preload the mouse with that, then you can see we're getting the control mice, lots of damage. Preload it, very good protection. If we infuse the dimethylmalonate at this stage, there's no protection. What's happening is that we think it's getting in, but it takes longer to hydrolyze and to accumulate enough of the active drug. So we thought, well, that's unfortunate. We can still use that for various other types of interventions, such as elective surgery or something like that. But what we want to be able to do then is to see, well, how are we going to get around this thing to make it a good drug, given at this point here? So what we did was to start, did some collaborations with a chemist. We started with the dimethylmalonate, which is slowly hydrolyzed, and we tuned the hydrolyzability of the ester here by just lots of different bits here. And what we found was that we can actually make very rapidly hydrolyzed malonites that can go in to the heart and deliver malonate. And this was the most rapid one, and the diacetyl epoxy will be very familiar to people like them. That's widely used to get things into the cell. So this is very effective. We can give that at reperfusion and get protection. At the same time, a group in Barcelona published a pig study which said that malonate itself was protective. So we thought this must be wrong because malonate has got 2 negative charges. Okay. So as you can see here, 2 negative charges, it can't get into the cell. So we thought this must be wrong. So we gave disodium malonate the 2 negative charges here. And what happens if we give it prior? There's no protection. If we give it at reperfusion, though, it's very protective, far more protective than all the various esters we spent a year making and testing. So what's going on here? It shouldn't get in. So the question was, if we generate malonate inside the cell, there's a very effective carrier in the memory called the dicarboxylate carrier. It will whiz into mitochondria and it will inhibit suction dehydrogenase. But how does it get in? We know that these do not get across the cell. There's no carrier for them or anything like that. So what's happened? So here in Prague, a very good PhD student and postdoc, who's now just set up his own lab now in Manchester in the UK, found out what was going on. If we take a heart, a Langendorf heart just hanging there, and we infuse malonate, disodium malonate, double negative stuff, and we give it to the heart, nothing gets in as we'd expect. We measure the amount of malonate in the heart. But if we let the heart go ischemic for a few minutes before we infuse the malonate, the uptake increases. So the longer ischemic time, the more uptake of the malonate you get here. And this uptake is going through a carrier called the monocarboxylate transporter 1, which is highly expressed. It normally works to transport lactate and other ketone bodies. So it's a very good transporter for those and very highly expressed. And what you can see is if we inhibit it with this inhibitor of the MCT1 inhibitor, we block this uptake. So it's a carrier-mediated uptake into the tissue by this carrier here. But the carrier activity is only switched on during ischemia or following ischemia. So what's going on with that? Well, what we found was that it's actually the pH drops during ischemia. So the pH drops to about 6.5 during ischemia. So when you're switching, just like if you don't tell your supervisor, but if you forget to feed your cells and come in the next day, you'll see that the culture medium's gone yellow because they're checking out lactate from glycolysis, and that lowers the pH. Same thing is going on inside the heart. And what happens is the malonate has a pKa of about 5.8. So at low pH, it becomes far more of a monocarboxylate form here. And the monocarboxylate is actually a very good substrate for MCT1. Okay. So what it looks like then is that what's happening is only in ischemia when the pH is low and lactate is built up, you can exchange for lactate through the MCT1, and you get this monocarboxylate now, and it will go in. So what we have now is an understanding how it was getting in, why it was better than the esters, and also that if we lower the pH of the malonate, the uptake will be enhanced even more. So if we lower the pH, say pH 6, infuse that at reperfusion, we should get very effective delivery here. We do that in mice upon reperfusion in the LAD model, and we get very good protection against reperfusion injury in that situation. So that's very good. Can we extend this to strokes? This is one thing, one intervention we could do. This can be given as a therapy along with PPCI to treat heart attack. But the other option we'd like to consider is stroke, because we're thinking that this mechanism of ischemia-reperfusion injury is universal. The other issue is in heart attack, people usually recover reasonably well from PPCI. They may get subsequent heart failure a few years down the track. But the unmet need in stroke treatment is huge because the vast number of people who get a stroke, even if they have treatment as quickly as possible, they're still impaired very badly. So what we want to think is, could we extend this to stroke? So first off, does this suction accumulation occur in a mouse brain? Here we do a mouse model of stroke. And the brain is great because you've got a control half and a contralateral and a sacral side. And what you can see here is that you've got this control bit. If you let it go ischemic, this is the bit that's getting damaged upon reperfusion by putting together all the metabolic changes. But again, we can slice through the brain and do this imaging mass spec, and you can see the succinate building up dramatically on this side of the brain and being oxidized very rapidly upon reperfusion. So the same process is going on there. So, but of course, we need to treat at reperfusion. The standard of care until recently for treating a stroke was to use a clot-busting drug like tissue plasminogen activator. The idea being that this will inject this into the circulation, it will digest the clot, and you'll reperfuse the tissue around there by getting the clot out of the way. And so that's just what we would used to be done. But that's difficult to apply our kind of therapy to, because there you're slowly digesting the clot, and so the malate will be coming in drips and drabs into the tissue, so it may not work as well. However, a more recent way of treating stroke is called mechanical thrombectomy. So what you do here, the same way as we now treat heart attacks, is you put a catheter into the large vessels in the brain where the clot is, take that through, you put a little microcatheter through the clot, and then you can use as a stent retriever. So our idea is that we can incorporate, as a microcatheter can also be used to deliver the drug. So our idea to translate that to humans, if we infuse the malonate into the brain just before we pull out the clot, then we should protect this region here from IO injury. So Jordan Lee, a good PhD student, just finished his PhD in my lab and that of Thomas Krieg, my close collaborator, who does the animal studies. He set up a system where we can mimic this in a transient middle cerebral artery occlusion method. What we do is we put in this occlusion suture, which will block the blood flow, have an infusion catheter over the top of this, and now we can infuse the malonate locally just as we pull out the blockage and reperfuse the tissue. So setting up this mouse model then was how we're going to see, would this work in stroke by getting the malonate into the tissue just at reperfusion. So let's see what the results would be. So first off, we just want to see, was the local delivery effective? And here what you can see is we just measure the malonate in the brain tissue. If we do a tail vein injection of disodium malonate, we get some uptake. If we infuse it locally, we get more uptake. If we acidify it, we get far more uptake. And if we block the MCT1 in the blood-brain barrier with an inhibitor, we block this uptake. So again, we're actually getting uptake through the blood-brain barrier into the tissue and getting target engagement, which is a major issue that we need to address in any drug that's trying to get into the brain. So, then is it actually doing anything? Well, we can use MRI 24 hours later, and what you can see here is that the saline on this control saline infusion, this is the side of the brain that's gone white, and that's showing that it's actually damaged. Here, you can see the white area is far lower. Because it's mice, we do MRI, T2-weighted MRI imaging, because that's what we would do clinically in the patient to just show how we can translate that. Because it's a mouse, we can also do histology, and you can see the dead area here, far less in the acidified disodium malonate treated brains here. Here we show the quantification of that. Here you can see that MRI, got a large volume infarct, and it dropped with acidified disodium malonate, and again measured by histology, it's dropped as well. And here, in parallel with those mice, we can do a rough indication of neurological outcome, the Bedison score, where 5 is very badly impaired and 0 is not impaired. And what you can see is the Bedison score is low in the green ones here, where they were treated with acidified disodium malonate, but it's higher where they were untreated, the black ones up here. So we've got a trend to better outcome, and also a lower infarct size in these mice here 24 hours later. So what we have then here is we've got a good indication that we can actually now develop treatments for stroke and for heart attack by infusing acidified disodium malonate. We get uptake here, and this goes into the dicarboxylic carrier, and we break down these, we block oxidation at cytosine dehydrogenase, and it will block the sequelae here of ROS production at this point here. So this is very good that we've found for the potential drug. At the moment, we're in the set of a company, we're doing the seed funding to try and see could we actually get that to work. So translation of this kind of thing is quite challenging. So you probably know that But this is actually, to illustrate how difficult it is that this is outside Cambridge as a cemetery. And in this cemetery, this is where all the startup companies go to be buried when they fail. And if you go in at night with a torch and you shine on the gravestones, what you'll see in all these buried dead companies is that, well, it worked in mice. So we're now very keen to go from the mouse stuff into large animals and humans trials as soon as we can, just to see, are we going to get any protection in humans, which would be our goal. And a lot of the work, so my work is done very closely with Thomas Creed here, who is a clinician cardiologist and pharmacologist. And what happens is, as well, we do the chemistry with Richard Hartley. He does a lot of the mitochondria targeted chemistry here. And then Kieran did a lot of the work on the heart that I presented here. And Jordan did all the stroke work that I presented here. And Bob and Abigail in my lab and in Bob's lab. There's a joint PhD program between the NIH and Cambridge. So she did a PhD back and forth with the NIH and Cambridge. So thank you very much for your attention. I hope that's been of some interest.
Thank you so much, Dr. Murphy. That was very, very interesting. And I'm going to pass the mic around for anybody who needs to ask a question. And I'm going to ask the question first. So you mentioned at the end, you showed the, I guess it's the disodium malonate was the compound that did you do the disodium malonate in the cardiac reperfusion as well? And did it have the same, yes. it all had the same effect? Yeah, okay.
It protects nicely in the heart.
Okay. And then I guess maybe maybe it's an obvious question or not an obvious question. What's the downside? So you showed that if you added it, obviously, like you said, before PCI, great. If you know you're going to have a heart attack, that's excellent. But at PCI, there's certain limitations with, timing, et cetera. So if we were to think about, especially with thrombectomy and other PCI, other types of therapies, is there a downside other than lack of efficacy? Is there harm at all to trying these kinds of things versus if you were to have this malonate species enter the cell and there sort of wasn't an accumulation of succinate, what balance would it throw off potentially? Is there any downside?
Yeah, the downside would be that it would be inhibiting succinate dehydrogenase, and that is a major part of the TCA cycle. So it would have some negative effects if there was sufficient there. However, it's a surprisingly safe drug. Largely because if you do it intravenously or intraarterially, it's not taken up into cells at pH 7.4.
Right.
So it'll stay in the circulation. And there is actually a specific dicarboxylate carrier in the kidney. And so these are cleared from the circulation with a half-life of about 30 minutes.
Right.
In rats and pigs. So it'll be washed out fairly quickly. Also, it's a competitive inhibitor. So what's going to happen is that it's not going to irreversibly inhibit secondary hydrogenase. So we find it's quite hard to kill people, or pigs or mice at least, with malonate intravenously.
Right.
So we think it's reasonably safe. It should just be taken up either when we do an acidified form locally where it's injected, or taken up by the ischemic zone.
Exactly. And then just before we pass on the mic, thinking about not a healthy reperfused heart, but maybe a diabetic reperfused heart, or one that's, in end stage or late stages of heart failure, where all of these metabolic pathways are perturbed, they're not perfect.
Yeah.
Any insight there?
As far as we can tell, because you're usually, because most of your work has been done on we like testing heart attack drug on this young audience.
Right.
Because you're not likely to have a heart attack. They'll tell me old people like me have a heart attack. So what we do is we also extend this to old mice and fat mice.
Yeah.
Old fat mice.
Yep.
And it's still protected.
Okay.
So but that's an important thing, that you're doing experiments on, doing all your experiments on a 25-year-old.
Right. Yep. Okay, great. King Women.
Thank you so much, Dr. Murphy. Great talk. So I'm just wondering how the drug was administered to the heart before the start of the reperfusion. Was the perfusion not completely stopped during ischemia?
Yeah. So what's happening when we're doing it, we're infusing it prior to ischemia. So you see it's infused over about 5 minutes or 10 minutes, and you start 5 minutes before you block off the artery, and so it's reperfusing around into the heart, and you keep it going afterwards, so it's still some loaded up. So that's when we're preloading it. But when it's at reperfusion, you're just infusing at the same time as you're on time.
Oh, you mean at the time of the reperfusion?
Yeah.
But the figure seems to show maybe 2 or 3 minutes before the.
But we start. That's the window of reperfusion.
Oh, okay.
It's not a bolus. It's a reperfusion over a few minutes. So we start before reperfusion. We start the pump and then we untie.
Okay. Thank you so much.
Thank you so much for your talk. Following up on what Dr. Rayner asked, I wanted to know the effect of malonate on skeletal muscles, which are known to have a slightly lower pH and also a highly dependent on aerobic respiration. So I want to know, is there something you looked at or you will planning to look at?
We haven't done that yet, but it's something we're keen to explore. It's universal. But muscle obviously is quite resistant to, or more resistant to ischemia-reperfusion injury.
Okay, any other questions from the audience? Marcelo?
Amazing presentation. My name is Marcelo. I work here as an associate scientist. The problem with reperfusion is that you show that oxygen travel much faster than any drug. And there has been some hypothesis that maybe reperfusion can be solved releasing slowly the oxygen, so they match the malonate intake. Have you considered hydrogel delivery so slowly oxygen can be recovered, or any other engineering approach?
I think it would be interesting, and I would think they would be complementary. But I think the whole idea of reperfusing with low oxygen could be beneficial for a number of reasons. One, it would slow the reactivation of mitochondria a little bit. Though, but bear in mind that it might slow the ROS production, but the Km of cytochrome oxidase is extremely low. So you just with a few percent oxygen, you're saturating cytochrome oxidase. But it would potentially impact on the ROS, because that's the second order process between the donor and the oxygen. So if the oxygen is lower, the ROS production rate will be lower.
Wonderful talk. Thank you, Mike. Yeah, I've read all your work, and it's just such a pleasure to hear you present all of this. It clarifies everything so well. And so such great translational research. Now, can you comment on, in vitro models, and if there is, to what extent is there use for, say, modeling some of these things in H9C2 cells, ischemia, is there room for cell biology characterization of affected processes, whether it's mitophagy or ferroptosis?
So, yes, we did a paper in Redox biology 2 years ago. What we showed is that most in vitro models of ischemia-reperfusion injury are useless because they don't really mimic ischemia. So obviously for us, the marker of ischemia is succinate. And if you do a normal low oxygen incubator, 1% or 0.1%, the succinate doesn't go up because of that very low Km of cytochrome oxidase. So it keeps going. HIF will be way up. Oh, sorry, I'll electrocute myself. So HIF goes shooting up, but the Km of that is way higher than for cytochrome oxidase. So mitochondria are activated by very, very low amounts of oxygen. So the only way, but what we did then, we set up a model using an anoxic chamber. So use a proper anoxic chamber that's used for protein chemistry, where you strip out all the oxygen chemically from the airlock to come in and out of that. So, and in that situation, in adult cardiomyocytes, we can get huge accumulation of succinate. We get reperfusion injury when we just do the reoxidation by taking it out of the chamber. We can see we see all those things in those cells.
Yeah. Great, thanks.
another question. Less in the, so thinking about not complete ischemia, not total occlusion, but sort of reduced oxygen, thinking of just this lack of, low perfusion. And I don't know if that would be considered sort of just smaller areas of ischemia that are the same pathology, or is it sort of variable? Is it just starving a percentage of oxygen? And in which case, does, again, sort of same question, can we solve some of those issues? It's not going to help the immediate changes in succinate because they're not immediate. So do you see any role in sort of a chronic ischemia kind of model?
Possibly, but again, I think it's quite the difference between low oxygen and no oxygen as far as mitochondria is concerned is absolute.
Right.
So if there's enough oxygen to keep it ticking over, then the electrons are going to go to oxygen rather than to fumate onto succinate. Yeah. So if you give them an alternative path, they're going to take that. Yeah. So I think unless there were local areas that were actually ischemic,
right,
then I think that we haven't explored that. My suspicion would be that an infarct area or might have areas of true ischemia.
Right. And then sort of the sequelae of those dying and dead cells, et cetera. So perhaps there is a role in those individual localized areas to improve the tissue area. Okay. Any other? Oh, there we go.
Thank you very much for your talk. I'm a student, so my question may not be super relevant.
No,
but
pretty best question.
So when you just started your presentation and the level of succinate and all those, that reminded me of in clinic when a patient is administered, they do check the levels of troponin T in their blood to kind of see if there is any cardiac injury or not. And I was just wondering if what you found can be more like also diagnostic application. I do understand because it's like within the 2 minutes, so it may be like too long, but I know like people also like do follow up to see if it is an ongoing cardiac injury or not.
No, it's a very good question. So a couple of things there. If we could use succinate diagnostically to pick out an ischemic zone. So we've tried ways of exploring that by MRI and by various other things, hyperpolarized various substrates. Or load them at C13. But the problem is the sensitivity of most of those techniques are not high enough to see the succinate accumulating in our hands. So we're still keen to do that. So apart from doing a biopsy, which we do in various, when you put a heart on a rig or a liver on a rig, then you can do those situations, but not so practical for a patient, really. But what we have seen is that in humans, we did a collaboration with guys in Oxford, and what they were doing is they had a catheter into the coronary sinus, and they were sampling blood at the same time as doing PCI. So what they were looking at then is they were sampling the blood as it was coming through, as soon as it was perfusing through, and we did see an enhancement in succinate, because half the succinate gets oxidized, half gets washed out. So some was being washed out and was accumulating, and we could measure that in that situation. But by the time it goes peripherally, it's diluted down in the 4.5 liters of blood. So again, diagnostic, it was more a proof that it was happening in humans, but it was harder to see how we could use that diagnostic.
Okay. Thank you all so much for the discussions, and thank you, Dr. Murphy, for this wonderful. I don't think there are questions online. No, I don't think so. I think all our online people are question free. So thank you again. Thank you for everything, and thank you for accepting our invitation to visit us. Thanks, everyone.